Okay, next topic in the homeostasis unit is gene expression. This is a short one. We've already talked about this in another lecture, but it never hurts to elaborate and review. It turns out embryology is just an incredible cascade of differential gene expression. And it turns out that observable cell differentiation, that is the process by where cells become different from one another in embryonic development, that results from the expression of genes for tissue-specific proteins. So, like kidney cells express different proteins than liver cells, than bone cells, than brain cells, than pancreas cells, etc. So, the control of one gene by another gene during development is called induction. So, one gene can induce another gene to do something. Induction of transcription factors, which are proteins that regulate the transcription of another gene, is crucial to embryonic development. Master control genes have been discovered that are known as homeotic genes. Evolutionary developmental biology reveals that many radically different animal species have amazingly similar homeotic genes. Humans and fruit flies share a lot of homeotic genes, which is kind of surprising because they're tiny and they're bugs, and we're big and we're not. In Drosophila, which is the genus of the fruit fly, the homeotic genes have a 180 nucleotide section called the homeobox. The homeobox specifies a 60 amino acid region called the homeodomain. Very similar domains called Hox genes are found in mammals and humans. Similar genes are found in plants, indicating that this embryonic control system evolved very early and was very important, and that's why it was conserved in evolution. The homeotic genes control the production of all kinds of transcription factors that regulate the timing and mechanism of other genes of embryonic development. Cascades of transcription factors turning genes on or off cause pattern formation, meaning like legs and arms, torso, ribs, segmented spinal or vertebral column, etc. Basically, temperature and the availability of water, which is an environmental stimulus, determines seed germination in most plants. Genetic mutations have been shown to definitely cause abnormal development. Genetic transplantation experiments support the link between gene expression and normal development. In other words, the current theory is in order to have a normally developing embryo, you have to have a normally expressing gene set. Any aberration in gene expression during development is going to be bad for the embryo. Genetic regulation by microRNAs plays an important role in the development of organisms and the control of cellular functions. Yes, RNA is not only for making proteins, it's also for controlling DNA. So it turns out that in Drosophila, most homeotic genes that have been discovered with the homeobox are associated with development. The homeobox encoded homeodomain is a protein binding site that binds to DNA as a transcription factor. So in other words, the homeobox is the part of the transcription factor that actually binds to DNA. Hox gene mutations produce odd body forms in animals. Varying expression of Hox genes along the body axis is what produces the different segments in the fly, and it's the reason why the thorax has wings but the abdomen does not. Plants do have homeobox genes, but they do not appear to work as master regulatory genes in development like they do in animals. That brings us to the fruit fly here. I believe this is Antennapedia. This is a fly that instead of having antenna on its head, it actually has legs growing out of its head because of a Hox gene mutation. Anytime you interrupt the interaction of different Hox genes, you're going to mess up the pattern of body formation in embryonic development, and that's not going to be a good thing for the embryo. Apoptosis, this is the last thing to talk about with gene expression. Very fascinating concept of cell suicide. The reason a foot has 5 different toes, or the reason the human hand has 5 fingers, is because the cells in between the fingers died. Your fingers did not develop as like root growths coming out of your palm. Instead, your hand developed as a paddle, and then you programmed the cells in between the fingers and between the fingers and the thumbs to just die. And when they did, that left 5 separate digits on your hand. So apoptosis is programmed cell death due to cell shrinkage, nuclear disintegration, and cell death. In mammals, apoptosis proteins called caspases cause mitochondrial leakage and the destruction of important enzymes in the cytoplasm. Cell death between fingers is important to the formation of the tetrapod hand and foot. We were talking about that already. Apoptosis is also important in the normal cellular prevention of tumor formation. So it's an important phenomenon to actually tell cells to do extreme things for the life of an organism. And apoptosis is pretty extreme when the cells here, here, and here just died in order for all the other cells to live. Pretty amazing genetic control there. And now we'll go on to the next lecture, which is photoperiodism.